Thanks. Let me first start by thanking Ralph and the Origin Institute for inviting me here, both for the conference and the opportunity to give this public talk. As Ralph mentioned, I'm at NASA, and so I'm interested in planets, obviously. But the planets that I'm interested in are those planets that can tell us about life. Obviously, we can learn a lot about life here on Earth, but it'd be nice to have another example. And that's what I'm interested in, where we find another example. And as I'll show you tonight, Mars is probably the other planet that's the most likely to tell us a story that's somehow related to life and its origin. So what I want to talk about tonight is searching for life on Mars. Where did we go? What we look for? And I want to use examples from Earth as a way to show how we are proceeding in trying to understand how to search for life, looking for it in extreme environments on Earth. And in particular, I want to talk about the Antarctic, dry valley lakes, the permafrost in the Arctic and the Antarctic, and also the Atacama Desert. These are the places on Earth. Well, these are my three favorite places. Whenever I get tired of the office politics, you know, and you just got to get out, that's where I go. I want to first start by saying that what I'm going to the talk I'm going to give is a compilation of work over many years by many people. I'm just the spokesman for it, and obviously they get thanks and acknowledgement. I remind myself who I am and where I am, and my email if you have comments that you're too polite to say in public. Let me start by addressing the question, why is life on other worlds interesting? Why do we care? Why are we having a conference on astrobiology? What is the real question? What are we really looking for? Well, I think the answer to that question is what we're looking for is an answer to the question is life common in the universe? Are we alone? That's the big question that astrobiology seeks to answer. And the way we're going to answer that is by finding a second example of life. As I'll show you, and as probably most of you know, there's only one life form on this planet, one example of life. All of us, all the organisms on Earth are related. They're all really the same phenomenon. Life here is singular. So we don't know if we are the only life in the universe or if life is common. To answer that question, we have to find one more example. So that's the first step in understanding the cosmic nature of life, is to find another example of life, a separate example, what I call a second Genesis. We, of course, are the first Genesis. We always consider ourselves first. We're looking for a second Genesis. And that would allow us for the first time to do comparative biochemistry, and it would answer the question that life is common in the universe, just finding one other example, particularly right here in our solar system. And I've added my own little editorial comment there. Yay, I think that would be a good thing, that if we found out that life is common. And we'll also learn some stuff about early planetary environments and the origin of life on Earth and so on. And those may be interesting. You may do a couple papers on them. But the real fundamental question is there life out there that's separate from us? So that's why we're searching for life. Why Mars? There's really three answers to why Mars is the target. One is that we have on Mars, as I'll show you, evidence for water in the past, liquid water. And we look around on life on Earth, we see that the common ecological requirement for life on Earth, if you could even call it the defining ecological requirement for life on Earth, is liquid water. So we have evidence for past liquid water on Mars. We see an atmosphere with carbon and nitrogen in it. And most importantly, we see on Mars the potential to find evidence of that life. It's a cold, static world. It's been that way for a long time. So if there was life there, we have the chance to find evidence of it. And that's, in a sense, my job description is not literally go there. I'm too tall to be an astronaut by a lot. But to go there robotically and with future astronauts and search for the evidence of that life to determine if it was there and if it was a second Genesis. Okay, so that's why life, why Mars. We're making good progress here. So Mars, let me just real quickly tell you what we learned about it and what we're learning right now. It started when Viking landed on Mars. That's when I got interested in the planet. Viking landed and showed evidence for water on the surface. You see these fluvial features in the lower right, and you see frost out on the surface. That's my favorite picture from Mars. The Viking image looking out on a cold morning, and you see the white stuff on the ground. That's frost. We know it's water frost and not CO2 frost because it's too warm to be CO2 frost. And you can see the antenna talking to Earth. It's just a nice coming together of the spacecraft technology, and the most interesting thing we know about Mars, that it has water, and the evidence that that water was liquid in the past. It's not liquid now, but it was liquid in the past. And the other point is the atmosphere of Mars contains all the elements needed for life. CO2 atmosphere, nitrogen, argon. Of course, it's different from our atmosphere. Our atmosphere has nitrogen and oxygen. But keep in mind that that oxygen in our atmosphere is only a recent product, a pollutant, really, created by life. Originally, our atmosphere was CO2 dominated, we think. And human beings in their wisdom are getting rid of that oxygen pollution and bringing back the CO2, restoring the Earth to its environmentally correct original condition. And many life forms will thank us for that, by the way. So this is a perfectly good atmosphere for life, if you're the type of life that's like a plant. Right now, I want to go a little more about what we know about Mars and what we're learning from the missions that are going on right now. There's 4 missions operating on Mars right now. It's really a great time to be interested in Mars because of the success of these missions. We've had a lot of failures, but we have 4 missions operating right now. And it is true that we send more missions to Mars. In fact, we crash more missions on Mars than we send to the rest of the planets put together. And the reason is Mars is the planet that's going to tell us a story, if any of them will, about the origin of life. So there's Mars Global Surveyor, Mars Odyssey, the 2 rovers on the surface, and a special note is the Mars Express, which is the first successful non-US mission to Mars, and hopefully, which will start a pattern that will continue as more and more nations and more missions to Mars are going. So I'm going to real quickly tell you, what have we learned for all this effort? We got 4 missions. What are the highlights? From the point of view of astrobiology, what are the highlights from these 4 missions? So just real quick, what have we learned? From Mars Global Surveyor and Mars Express, we're seeing better evidence that there was water on Mars and that that water flowed stably on the surface. This picture here, I would say, is the poster child for water on Mars. This is a canyon, the Edie Canyon. The canyon is about 2 kilometers across. You can see that the canyon snakes back and forth. Up in the top right, you can even see the channel of the river that's on the bottom of the canyon, or was on the bottom of the canyon. It's like the Grand Canyon. If you look down the Grand Canyon, careful to avoid falling, you can see the Colorado River. You don't have to be a geologist to realize that it took that little river a long time to carve the huge canyon. And that's what we're seeing on Mars. We're seeing that little tiny river channel on the bottom of the canyon. And the fact that it's snaking back and forth argues that this has been slow, stable flow. This wasn't a giant flood that came gushing out from a broken ice dam or something and carved the river very quickly. Some of the features on Mars could be explained that way. And for a while there was a big debate about maybe there was never water on Mars for a long time. They were just these catastrophic floods that would come in a few days and they'd be gone. This is evidence for stable liquid flow, and the only liquid that really can be an explanation for that is water. The other liquids people have talked about, wind, lava, so on, don't fit this. So the Global Surveyor and the Mars Express are showing us better evidence for water on Mars in the past. We have never seen any liquid water on Mars today. It's only evidence of liquid in the past and ice and vapor today. Where's the water? Most of it, well, that's what Odyssey is telling us. Odyssey is confirming the supposition that we had that the water on Mars is frozen as ice in the polar regions. This is a map showing ground ice on Mars. You can see the permanent ice-rich ground is the blue in the north and south region. So we're having a story that's coming together pretty nicely. There was water on Mars. It's now frozen as ice in the polar regions. The rovers have confirmed that there was water on the ground. The data so far has been from orbit. Data from the images and from the gamma ray spectrometer are from orbit. On Mars, on the rovers, we have the same confirmation of water on the ground, and that takes 2 forms. One is morphological and the other is geochemical. Morphologically, we see in this kind of image, this is a picture of a crater wall that Opportunity landed in at Meridiani Planum. You can see the layering indicating, or one interpretation of which is layered, deposited in water. And then measurements were made. You can see the 2 circles, high and low in the rock. And the rover measured the salt concentrations high and low, and that's shown in the next slide. The highest salt, the highest sulfur, was found upper regions of the surface. This is just what we see in deserts. In Death Valley, for example, water carrying salts percolate up to the surface, evaporate, leave the salts behind on the surface. We're seeing that same sort of desert crust on Mars. So with these rovers, we're getting on the ground evidence that there was a liquid and that that liquid was water. So there's no clear limit to how long those rovers can rove. So they're still growing strong after more than a year on the surface of what was supposed to be a 90-day mission. Okay, so salts. We found, trying to remember where I am. Oh yeah, last evidence that the rovers gave us for water were these so-called blueberries. These little round things that were neither blue nor were they berries, but they're called blueberries. They're concretions, and they form when water has some, in this case, hematite in solution, and it's saturated and precipitates out, forming concretions. You see them in sandstone, for example. Evidence for liquid water as well. So the case for water is good, and the image that people sometimes draw is one like this. This is an artist's conception, obviously. It's not a photograph of Mars. Artist's conception of Mars, what it might have looked like 3.5 billion years ago. And the artist's conception is actually based on the amount of water the geologists think Mars must have had to explain the features that we see. A layer of water equivalent to 500 meters over the entire planet. If you let that water flow on the current Mars, it would flood the northern plains and create a scene that looks just like this. Well, when you look at this picture, does it remind you of anything? Well, it probably reminds you of Earth, right? And that's why, this is why Mars is so interesting. Not the Mars today, but the Mars of 3 billion years ago, when Mars maybe looked like this, when we had 2 blue marbles in our solar system. And in fact, one good question is, well, what happened? Why did we lose one of our marbles? What happened? Why do good planets go bad? And with Mars, the answer is really quite simple. It's not because it's so far from the sun. That's what I learned in kindergarten. It tells you how long ago I was in kindergarten. That Mars was cold because it was further from the sun than the Earth. That's in fact not the important distinction between Earth and Mars. The important distinction between Earth and Mars is that Mars is 1/10 the mass of the Earth. Mars is very small. You have to be this high to be a habitable planet, and Mars is that high. Right? And why size matters? No plate tectonics because it's too small to support it, less gravity, no magnetic field. All of that resulted in Mars losing its atmosphere, both to mineralization, forming carbonates, for example, and lost to space. No atmosphere, no greenhouse blanket, gets very cold, all the water freezes. That, in a nutshell, in 25 words or less, is the history of Mars as we understand it. And plate tectonics is probably the big key. It's now what we can think of as our unifying theory of how planets work. It's the answer to the problem in planetary science called the Goldilocks problem. Why is Venus too hot, Mars too cold, and Earth just right? Hence the Goldilocks problem. And the answer, we think, is plate tectonics. Venus doesn't have this cycle because it's too hot to have water. Mars does not have the cycle because it doesn't have plate tectonics. And so the cycling of carbon, which on Earth is done by plate tectonics, doesn't happen on Venus, doesn't happen on Mars. Those planets don't have the complete cycle the way the Earth does. We'll be able to test this model when we find planets around other stars that are like the Earth and Mars. But for now, this is how we think the planets work. But there's a paradox about Mars that I want to come to. So Mars was warmer and wetter in the early period, but it wasn't that much warmer. And we see this on the surface. Here's one of my favorite places on Mars, Gusev Crater. The Opportunity lander, the Spirit rover landed right on that red X. Gusev Crater is a big crater, 160 kilometers in diameter, and from the bottom you can see what looks like a large river that flowed into the crater. You can even see what look like delta deposits at the mouth of the river. So here you have a large river. That river is about 2 kilometers in diameter. So that's the size of the Mississippi at St. Louis, for example. And there's a large crater, 160 kilometers in diameter. A large river, a large lake, and yet look at the terrain around it. You see what look like moon craters. Obviously inconsistent with an Earth-like environment with rain. So we have this paradox of Mars, which it looks like it had water, it looks like it had rivers, it looks like it had lakes, and it looks like it never rained. And so how can we reconcile that? Well, this is another bit of evidence that Mars looks like it never had rain, which is the evidence that the surface rocks are pristine volcanic rocks. I'm not going to dwell on that, but for those of you who are interested in it, you can go to the tests top 25 science results web sheet and see this discussion. We have evidence that Mars was a cold and wet place. And on Earth, where do we see that? Well, you might have guessed from my introduction, Antarctica. And in Antarctica, we find environments that are like what we think Mars was like. It was not like Miami Beach. It was like the dry valleys of Antarctica. This is a Landsat view looking down on the dry valleys. They're the largest ice-free region in Antarctica, the region down here. If you look up onto the upper part of the picture, you can see McMurdo base, the little black dot on the icebreaker track coming in. The dry valleys are this dry region. They're dry because of a mountain range, the transantarctics, that blocks the flow of ice into the valleys. It never rains in the dry valleys. It only snows. And the snow there is about equivalent to the rain in dry deserts on Earth. But it never rains. It only snows. One big difference between the dry valleys and Mars is the pressure is higher there. The pressure is high enough that liquid water is stable in the dry valleys, but it's not stable on Mars. On Mars, a glass of water would start boiling away and freezing simultaneously. Water on Mars behaves the way dry ice does on Earth. It doesn't have a stable liquid phase because the pressure is so low. But in Antarctica, it is stable, and that's the big difference, but there's still some important similarities between the dry valleys. One is we see there rivers and lakes without rain. Here is the Onyx River flowing into Lake Vanda. Lake Vanda is about 5 kilometers across. The Onyx River is the longest river in Antarctica. It flows about 2 or 3 weeks a year, and usually it's about 1 meter or so wide. Some years it's bigger and flows longer. Some years it doesn't flow at all. And it flows only in the summer, obviously, when temperatures get above zero for just a short period of time. And it flows from those alpine glaciers that surround the valleys. Snow that falls on the shores, on the sides of the valley, just evaporates and provides essentially no liquid water. There's very little, essentially nothing growing in those soils, and there's no weathering due to that. But that large lake has water under it and an ice cover about as thick as this room. But underneath that ice cover, there is water. The cycle there is rivers and lacks. I guess that lakes there should be an E in there. Rivers and lakes without rain. It's a snow-based hydrological cycle. Evaporation, snow, glaciers melting, flowing into an ice-covered lake. This could work on Mars if the pressure was higher. The only thing we'd have to do to Mars to make this kind of system work is not warm it up, but make the pressure higher, maybe 10 times higher, and a system like this could work on Mars. So we think that this is what the hydrology that we're seeing on Mars, the rivers and lakes we see on Mars, is these kind of rivers and lakes like the Antarctic. And we can model that. I won't go into detail, but we can write down the equations of the ice cover and the environment that's providing it, and then we can apply it to Mars and show that Mars could have had a period of time when it had ice-covered lakes, even though it was no warmer than it is today. So why is this interesting? Well, we go down to study these lakes. This is our camp. This picture was taken on Halloween, actually, which is why we're flying the Jolly Roger. We had a bowl of candy out. We were waiting for trick-or-treaters, but none of them came, so we ate the candy ourselves. You can see that's our dive hut, the dark hut, and then there's the generator shed, the lab, and the facilities. There's a little narrow wooden box there, and the big white glacier is our water supply. And from that, we go out onto these lakes and we melt the hole. As we melt the hole down, the hole is dry, and this is a view up the hole as we're melting down. And when we pop through the lake, the water comes up. So there's water underneath the ice cover, and then we get divers, get volunteers, tied snugly, of course, lower down the hole. The water's exactly at zero. This is the view looking up. Why do we do that? Well, because on the bottom of these lakes, in this otherwise essentially lifeless dry valleys, there's algae growing underneath the ice, in the water, essentially protected by that layer of ice. That layer of ice acts like a blanket that keeps the water liquid year-round. And the organisms live in the water. 0 degrees water is perfectly fine for these algae. All we find in this lake are algae and bacteria and simple life forms. We do not find insects or fish or anything like that. Just primitive life forms, algae, and scientists from NASA coming down to study them. Well, that's what we're thinking could be on Mars. Go back to this lake, Gusev Crater. The evidence that this was very cold while there was water flowing here is what leads us to think that the best analog for this lake is a place like the Antarctic. But just like there could be life in Antarctica, or there is life in Antarctica, there could have been life in this lake. So one of the goals we have is to dig down in these kind of sediments and look for the fossil remains of this life, of the green slime that we're finding in the bottom of Antarctic lakes on these Mars lakes to see if there's any life there. And that would be very interesting. Another possible target that's recently been discussed is this evidence for what the Mars Express team thinks is a frozen ocean. This is a picture that they say is the remnants of a frozen ocean. And indeed, those rafts look like ice rafts. If you ever fly across northern Canada, you can see these even in the summer, these ice rafts. So it could be a frozen ocean. There's some debate on that. But again, life in the polar regions on Earth shows that life could survive there, and maybe searching for fossil evidence is good. But I want to now talk about beyond the water. We found the water. We have a good model for the water. Water on Mars, we have the water in the Antarctic. Now what do we want to look for? And I want to talk a little bit about targets, possible targets for searching for life beyond the water. And I want to focus on targets that can answer the question that I started off with. And the question was there a second genesis of life on Mars? And so the first thing I want to say is that fossils, while they would be interesting to find, are not a way to answer that question. When I first started getting interested in life on Mars, it was assumed that if there was life on Mars, it had to be alien. It had to have started separately from Earth. Life started on Earth, life started on Mars. They're different planets, got to be different life. But we now realize, and I think that's in the next diagram, that life on Earth and Mars may be the same. Actually, this diagram is actually to reinforce this philosophical point that we're looking for something different. This is a figure that Scott Sanford put together for a paper for the Journal of Irreproducible Results, in which he compares apples and oranges. You say, well, why would you want to compare apples and oranges? Suppose you were doing your thesis or your honors project on fruit. You'd want to compare apples and oranges. If all you had was oranges and you're trying to describe fruit, you would have a limited view of the full diversity of fruit. You'd like to have apples. In fact, you'd like to have bananas. You'd like to have tomatoes. Tomatoes, biologically, are obviously fruits, even though they're in the vegetable section of the store. At least in the U.S., that's because in the 1800s, the Supreme Court declared tomatoes vegetables. The Supreme Court has done other things, too, that maybe are equivalent to that. But so the situation we are on Earth is that we have one example of life. In fact, here is the one example of life. This is the tree of life. It shows all organisms on Earth, and it shows that they're related to each other. This is Carl Woese's 16S ribosomal RNA tree. And this is a genetic tree. We've known for hundreds of years that all life on Earth is made out of the same stuff, that the hardware is all the same. What this tree shows is that all life on Earth has the same software. There's one operating system for all of the biological systems on Earth. Just like in the future, there'll be one operating system for all the computers on Earth. We'll all be using Macs. Well, that's the same, yeah, VMS even. This is interesting. I think it's one of the most profound results of science for the last 20 years, is the phylogenetic unity of all life on Earth, and what's more, the ability to root that tree and point to the location of the common ancestor, shown as a little red offshoot there. What we were looking for, in a sense, when I say searching for something different, is something that's not on that tree, something that is not related to us. Right? We know how to identify things that are on that tree with very high precision. What we want to find is something that is not on that tree. Now, as I said, we assumed for a long time that life on Mars would be alien, would be different. But now we no longer assume that. Wow, that was that woke me up. Now we no longer assume that, and the reason is because we have on Earth rocks that have come from Mars, like this one. the Allen Hills 84001. It was a rock found in Antarctica that came from Mars. We have about 20 or so rocks on Earth that came from Mars. Well, how do we know they came from Mars? They don't have little plaques on the bottom that say made on Mars. Right? What they have, though, is inside the rocks, there's little gas bubbles, and those gas bubbles have exactly the same composition as Mars air. Mars air was measured by Viking. Remember, I showed it to you, the third graph. Carbon dioxide dominated, nitrogen, argon. No other planet has that kind of air. Earth doesn't, Venus doesn't, Pluto doesn't, Goofy doesn't. None of the other objects in the solar system have that kind of gas, so it must come from Mars. And how did they get here? Well, we think they got knocked off by asteroids. This was a big surprise. The theorists could prove, until confronted by the data, that rocks could not go from Mars to Earth, and yet they're here. Further, we now know that these rocks could carry organisms from Mars to Earth, and those organisms could land on Earth and still be alive. So now, in fact, the default assumption would be that Earth and Mars are biologically interacting, or have in the past. Just the way that the continents on Earth biologically interacted and are interconnected. We would not expect to find a different second genesis of life in Antarctica or Australia, because we know that on Earth the winds carry microbes everywhere. Microbiologically, the Earth is a single place. Well, now we know that that may also be true in the planet. So the burden of proof, if we find evidence of life on Mars, the burden of proof that it's different, that it's alien, that it's a second genesis is on us. We have to show that it's different. We can't just assume that it's different. If we find a fossil, say we find a footprint or a fossil, that tells us that there was life on Mars, but it doesn't tell us if that life was our cousins from way, way back, or if it represents a second genesis. We don't need a footprint, we need a foot. Think of that show, Crime Scene Investigation. What they like to have is dead bodies, because then they can analyze them. That's what we're looking for on Mars, not a fossil on the bottom of a lake, although that would be exciting. We're looking for actual organisms, alive or dead. They can be dead. If they're alive, you just have to kill them anyway to do the analysis, right? Dissolve them in acid and look at their guts. But they need an organism. There's a big difference between a dead organism, a corpse, and a fossil, right? And that's what we're looking for. So that, where can we find corpses or fossils on Mars? Well, we look on Earth. Where do we find the oldest dead organisms or alive organisms on Earth that could be a model for Mars? The answer is in permafrost. One thing Mars has a lot of is frozen ground. I showed you the data that showed the ice-rich ground in the polar region. Well, if we go to the oldest permafrost on Earth, and the oldest well-dated permafrost on Earth is in Siberia. Here we find ice. This picture shows a cliff being eroded in the permafrost by a river, the Kolyma River, and you can see the drill pipe of the Russian drill. The top is the active zone, the surface, the first meter is the active zone, and then you can see the dark wedges of ice. That sediment is 3.5 million years old. We know the age from magnetic reversals, and we know it's been continuously frozen from the geomorphology of the ice wedges. With our Russian colleagues, we go back away from this cliff and we drill down and we find organisms frozen 3.5 million years and still viable. They're actually recoverable. Well, that's 3.5 million years. On Mars, we would be looking for organisms that were frozen for 3.5 billion years. Million and billion, they sound alike, but there's a big difference, right? You can ask Bill Gates, he can explain it. Well, so what we're now trying to find is older permafrost on Earth. And we think in Antarctica, here in Beacon Valley, underneath these tents, underneath that ablation till, there's ice. And there's some debate as to the age of that ice, but I think there's a good case to be made that the deeper layers of that ice is 8 million years old. That is now the oldest ice on Earth, 8 million years old. And in that ice, at minus 25 degrees, we also find organisms preserved and still alive. Now, 8 million, we're getting closer to 3.5 billion, but we're not there yet. So we have to extrapolate. So how do we extrapolate? Let me see if I can get to the next slide. It'll probably jump two at once. Oh, let me first explain how we do the drilling. This is a drilling rig. This picture is actually from drilling we've been doing in permafrost up in Ellesmere Island near Eureka, where we're also doing the same thing. drilling down. And there what we're trying to do is develop techniques to drill aseptically. If we're looking for organisms in a sediment, normally you collect them with a sterile tool. Well, you can't sterilize a drill rig. So what we do instead is we coat the cutting core with fake bacteria, 0.5 and 0.05 fluorescent beads that act like bacteria. So if we were contaminating our samples with bacteria from the cutter, these fluorescent beads would also contaminate it. And some work for some folks up at McGill working on this project together with us using the NASA Johnson drill. You can see that this is a fluorescent image, a picture of the fluorescent beads, and you see they only penetrate a 1/10 of a millimeter into the core, and the interior of the core is still clean. The moral of the story is we can use a drill rig and in frozen ground take a sample without using drilling fluids, and the interior of that sample is still clean and pristine, so we can recover microorganisms from that sample and be confident that they were in the ground. So we want to do that on Mars. Where is the ancient ice on Mars? Where would we go to drill down in ancient ice on Mars? Well, this picture, it's kind of complicated, but it's a map of Mars. North at the top, south at the bottom, and it shows where the old ground is on Mars, and it shows where the ice is on Mars. The old ground is indicated by the craters. Each little green circle is a crater, and you can see that the bottom part of the planet has lots of little green circles. That's the cratered old part. The north part of the planet is smooth. That's the young part. The ice regions are shown by the blue lines. Blue lines are the ice regions. So if you wanted to find old ice, go to the south below the blue line. But we learned something new recently from Mars Global Surveyor, which is the red and blue stripes. Those are magnetic stripes, extremely strong magnetic stripes. If you were riding your bicycle, or my bicycle, which has a little compass on it, across the surface of Mars where those colors are, the compass would swing north-south as you went up and down there. And that's puzzling because Mars has no magnetic field, no dipole magnetic field. Where did those stripes come from? The answer is we think that those stripes are left over from very, very early in Mars's history when it did have a magnetic field. These stripes may be the oldest thing we see on Mars, these magnetic stripes. And in fact, you can see where the giant craters, Hellas and Argyre, have erased the stripes around them. So where the stripes are present, that terrain has not been heated above the Curie point or shocked to pressures high enough to destroy the magnetism, the coherent magnetism in those areas. So if you were to say, where then would you go to find old, ice-rich ground that's been undisturbed by big impacts? Well, that X right there at about 185 west and about 80 south. You see that yellow X? That would be a good spot. That yellow X, as it turns out, is the crash site of the Mars Polar Lander in 1998. So we would land right there, maybe not on the same crash site, maybe not on the crater, but a little bit over, and drill down, and there we would have a chance of finding ice, frozen ground on Mars, that's 3.5 billion years old. And we would look to see if there was organisms in it. So would they be alive? Well, I don't think so. The temperatures are cold, so thermal decay is not important, but just background radiation from crustal abundances of uranium, thorium, potassium would have killed them. It's dead, Jim. But as I said before, dead is fine. In fact, dead is good because then we don't have to worry about whether they're pathogenic or going to cause infections or whatnot. The important distinction is that they are not fossils. They are corpses. We can analyze them directly. We can see if they are on the tree of life or not. What we're hoping is that they're not on the tree of life, that they really are alien life form, which opens up an interesting question. How do you detect an alien life form? The answer is we don't know. We know how to detect things that are on that tree, but we don't know how to detect things that are not on that tree. We have to figure that out pretty soon. I wanted to go back to how have we tried to detect life? We've only tried to detect life once, and that was on the Viking mission. Viking scooped up some soil and it analyzed it to see if there was anything in the soil that could grow when given essentially chicken soup. Add chicken soup, see if anything grows. That's what Viking did. The next thing is just a schematic of the Viking biology experiments. There's the chambers, soil was added, and then water and nutrients were added. What Viking found is that something was in the soil. There was something in the soil capable of consuming organic material. Something ate the chicken soup. But one of the instruments was an organic analyzer, and it found no organics in the soil at all. How can there be life without organics? So on Mars, what they found, the conclusion was that there was no detectable life, no detectable soil no detectable soil organics, and presumably something like an oxidant that's capable of consuming the nutrients. So one of the questions is, does this occur anywhere on Earth? Or ask the question another way, is there anywhere on Earth where Viking could have landed and not detected life on Earth? Any soil. Obviously, if it landed on pure ice, it wouldn't have detected life. But if it landed on dirt, is there any dirt that you can pick up on Earth and do exactly what Viking did and not find evidence for life on Earth? Very hard. There's no place that you can think of, but there is one place, and that is in the Atacama Desert that we sort of literally stumbled across. This is the Atacama. You can see this is South America in many colors. The Atacama Desert is located on the west coast of Chile and Peru. You can read too, I guess. Here's a view of the Atacama looking down, and you can see why it's so dry. This is a picture from the Hubble telescope looking down, and you can see why they're afraid to go back to Hubble. They could fall, go straight down and land in the Atacama Desert. You can see why it's dry. On one side are the Andes, a huge set of mountains blocking moisture from that side, and on the other side there's the cold Humboldt Current, which keeps the moisture availability down, and a coastal range that blocks the fog. And you can see that narrow stretch right there is the Atacama Desert, the driest place on Earth. It is so dry that we've been working in deserts for many years. We went down there and put in a weather station, and it recorded nothing for 2 years. I thought it had broken, so I went down there with replacement parts to fix it. It's just that it doesn't rain. It hadn't rained in 2 years. This is also a region where they mine nitrates, and this is a picture of the desert. And you can see where they had mined the nitrates. It's the only place where commercial nitrate mines. An indication that it was dry was these algae that grow under stones. In all deserts of the world, go to a desert, any desert except the Atacama, find quartz stones, the white stones, and pick them up, and underneath there'll be a green layer. It's kind of a fun thing to do if you're in a desert and sort of at loose ends. Go looking for white stones, and they're all colonized, and they're usually colonized by the same species, a type of cyanobacteria. In the Atacama, we find those stones, and they are not colonized. I think that's this figure to just convince you that we measured them, counted them. In the dry region where the rain falls below averaging 1 millimeter per year, the colonization drops essentially to zero. I don't want to go into detail, but the organisms that aren't there, that was the first indication of how dry they were. Like I said, we put in this weather station. We recorded 4 years of data before the station was stolen. And in those 4 years, we got one rain event. It rained once in 4 years, and it occurred at 1 o'clock in the morning, and it was 2 millimeters of rain. It was not detected or recorded by the normal weather station that was just a mile away. We detected it because we had a special electronic tipping bucket rain gauge that could record very sensitive rains. This is really the driest place we've ever been. To put it in rough numbers, it's 50 times drier than Death Valley. Really dry. And this is a diagram of why. It's blocked by mountains on both sides with, just for good measure, another blocking mountain range in the middle. It's really quite a interesting place. This is also where the sodium nitrate deposits were accumulated. As you may remember, these were mined by the Germans and the British, and they made bombs and threw them at each other. World War I and World War II, and until they invented the Haber-Bosch process, this was the main source of nitrates. So it's an interesting bit of history there. Well, what we did is we did a transect from the wet end of the desert to the dry end. And the dry end is this region about 100 kilometers wide. And the thing I want to focus on is the bottom diagram, which is the microbiological concentration. If you start at the wet end, you find that's in the soil, there's bacteria, just like in the Death Valley or the Mojave, 1 million to 10 million bacteria in every gram of soil. As you get closer to this dry core region, they suddenly just fall off by 4 orders of magnitude. So it goes from 10 to the 6 to 10 to the 2 or less. That's a log scale there. It's just like, it's equivalent to what happens when you look at a mountain and you see tree line. You go up a mountain, it gets colder and colder and colder, the trees stop, and there's just a sudden drop in the trees. It looks like someone shaved the trees. We're seeing the same thing here microbiologically as we go into the dry zone. So we wondered if there was also an oxidant like we saw on Mars. Unlike the case for Mars, which just put a generic soup together, we made two types of soup. A soup composed of L amino acids, which is what all life on Earth uses. We can call that the good soup. And a soup made of R amino acids, which are the mirror image, which organisms on Earth do not use to make proteins. All the amino acids in your body are L type. If you digested R amino acids, your body would not be able to use them in making proteins. Right, so life has a preference for one type. Chemically, they're identical. So an oxidant, something that's not biological, will eat both of them, and something that's biological will eat one and not the other. And what we found, shown here, is that in the core dry region, there is something in the soil that's eating the organics, but it eats the left and right-handed equally. You can see that down here, the ratio of the lower diagram, the ratio of the biotic to the non-biotic is 1. Whatever is in the soil at the desert in the core, lifeless region is eating organics, it's eating it chemically. Move down to the wetter end, and the biological response shoots up. We think that this then is a model for Mars. This is a lifeless place which still consumes organics because of chemistry in the soil. If Viking had landed right where that flag is, we would have been very surprised to see it there. But if it had landed there and done its experiment, it would not have detected life on Earth. This is the only place that Viking would not have detected life on Earth with the gas chromatograph or the labeled release or the Gex experiment, and it would have seen the same activity. It would have seen something consuming organic soup in the soil. So this is a diagram of deserts, sort of just a moisture versus life versus water. Most deserts that we're familiar with, like the Mojave and the Gobi, have about 25 millimeters per year of water or more. The Atacama is down at around 1 and even lower. It goes down 2 orders of magnitude lower on this two steps of 5 lower. And it gets into a region that we call Mars-like soils, where biology is no longer a force. It is so dry that life can't make it at all, even bacteria. Okay, I want to stop now with just sort of a last image, which is here, and leave time for questions and answers. The last image I want to leave you with is, up to now I've been talking about Mars' past. We think that Mars was wet. We think because it had water, there's a possibility that it had life. We want to go there and find evidence of that life. We want to see if that life was a different genome than Earth's genome. Why do we want to do all this? Well, one reason is to see if there was life, to answer that big fundamental question, are we alone? But there's another big fundamental question, I think, that we're also asking in our Mars research, which is, where are we going? Where are we going? And the questions are related. Are we alone, and where are we going? If there's no one out there, then that's a different destination than if the universe is full of life that we can go out and explore. Where are we going on Mars? That question translates into, does Mars have a future? Could we imagine a biological future? And if Mars has life of its own, could that future habitable Mars have its own genome? Another planet with its own genome, bring Mars back to life, both biologically and physically. If it doesn't have a genome, we could bring it back to life physically and share our genome with Mars. Either way, we could have 2 biologically rich habitable planets. And the more we study Mars, the more we see that it is a planet that was, in some sense, meant for life. It's just recently died, just a few billion years ago. We could do CPR and bring it back to life. How do you do CPR on a planet? Well, the fundamental problem in bringing Mars back to life is warming it up. Well, we know how to warm up planets. We're doing it on Earth, where it's not a good idea. But in fact, if we were to do the same thing on Mars, it would warm up, the water would melt that's trapped in the ice, Any CO2 in the polar caps or in the regolith without gas. And Mars could be once again a planet suitable for microbes and small plants and things like that. Could be very interesting. Let me stop there and maybe we have time for questions.